Rebecca Benezra, graduate of Northwest Yeshiva High School and now a rising freshman at the University of Maryland. So come on up.
Hello, everyone. I'm Rebecca Benezra, and I had the pleasure to work at the Lein lab at the Allen Institute. I want to thank everyone for being here. So my project was on pathological tau protein buildup in varying Alzheimer's disease severities. So first, a lot of background information. Alzheimer's disease, or AD, is the most common form of dementia, and it affects almost 7 million people in the US ages 65 and older. An important word to know is the word pathology, meaning the behavior of the disease. In AD, the pathology looks like a buildup of amyloid plaques and a buildup of pathological tau protein. These two things inhibit neuronal function, causing symptoms such as loss of memory, logical reasoning, language skills, mood and behavior regulation, and a loss of coordination. The Seattle Alzheimer's Disease Brain Cell Atlas, or CAD, is a collaboration between the University of Washington, the Allen Institute, Kaiser Permanente, and the ADRC to map the different cell types in aging and Alzheimer's disease. This will help us understand how AD affects different brain regions and cell types. They do this by collecting donors with a wide range of AD severity and use mapping to identify AD vulnerable cell types and how they degenerate both structurally and functionally. My project fits into this because we are continuing this project of mapping into the locus coeruleus, a part of the brain that hasn't been mapped before. The locus coeruleus, or LC, is located in the dorsal pons of the brainstem, and it's very small, smaller than a grain of rice. The LC influences many of our brain functions, largely due to its connection to so many different brain regions. It is also the primary producer in the brain of norepinephrine, which is an excitatory neurotransmitter that plays a crucial role in many of the functions lost in AD. Now, earlier, when I was talking about AD pathology, I mentioned that pathological tau, or p-tau buildup, is a major characterization of AD. Since the LC is one of the first places where this happens, the LC is very relevant when talking about early AD development. Now, our project. To continue the CAD project into the locus coeruleus, a lot of foundational work needs to be done, and we tackle some of that work. So first, we need to be able to locate the LC in our tissue, and once we do that, establish a method of analyzing the LC and its generation in AD. And the third thing, which is more our hypothesis, which is that we will find the established relationship between AD severity and p-tau protein buildup in the LC of our cohort of donors. This graph represents how we organize our donors. We group donors according to the Brock stages framework, which uses a measurement of p-tau throughout the brain to predict stages of AD severity. So as you go through higher Brock stages, you have a higher predicted AD severity. Now our method. So first we had to decide which tissue we were going to use for our project. And we did that by looking at the tissue and seeing if we could either see locus coeruleus within that tissue, or if we had a good idea of where it would be. We then sectioned using a cryostat, which is essentially a freezer that allows you to take very thin sections of tissue. In our case, only 14 micrometers thin. We attached the slides and then antibody stained with two main antibody stains. The first was tyrosine hydroxylase, or TH. TH would bind to the LC's neurons. The other stain we used was AT8, which binds to p-tau. This would help us measure the presence of p-tau within each donor. And finally, we imaged using a fluorescent microscope and camera. So this is an image of one of our AD donors with a high predicted AD severity. As you can see, let's use the pointer. Oh, doesn't work. Okay. The purple represents the TH positive area, therefore the LC's neurons, and the green represents the AT8 positive area, therefore area where p-tau is present. Using QUPath software, we annotated this image and the locus coeruleus as a region in red, as you can see, and within the region, all the Th positive cells in yellow. In this magnified image of the annotated image, you can see that there are quite a few neurons that come up as purely purple, meaning that they have very low p-tau presence. However, other neurons come up as either green or white, meaning that they have a heavy p-tau presence. And finally, our results. The first graph represents the percentage of LC area stained by AT8. And since we know area stained by 8 means p-tau presence, this graph really represents percentage of LC area with p-tau present. And as you can see, as we go through farther Brock stages, a higher percentage of the LC's area has p-tau present. The other graph, instead of looking at the LC area as a whole, it looks at each individual Th positive neuron. This graph represents the percentage of Th positive cells with AT8 positive area over a specific threshold, meaning the percentage of the LC's neurons that has p-tau present over a certain threshold. And once again, as you can see, as you go through higher Brock stages, therefore higher predicted AD severity, a higher percentage of the LC's neurons have p-tau present. Now our takeaways, and first our successes. So we were successful in developing a robust method for localizing and analyzing the LC in a cohort of donors with diverse severities. In addition, we were able to confirm our hypothesis, which is that we find that p-tau buildup in the LC of our cohort of donors correlates with AD progression. In addition, that we found an increase of accumulation of p-tau within the cell. And finally, our next steps. Our next steps would be to continue this experiment with more LC donors, therefore finalizing this step in the preparation of bringing the CAD project into the locus coeruleus. Thank yous. First, I want to thank Dr. Ellen Bogan and everyone who made this program possible, and I especially want to thank my mentors who carried me throughout this whole project. Very inspiring, and I really appreciated their help. I want to say that everyone who took part in this project is so inspiring in their clear determination and love for medicine and science. And I also want to just thank everyone who's here, both virtually and in person. So thank you very much, everybody.